Molecular biology behind the genetic material DNA. DNA is made of a long sequence of small units strung together. There are 4 basic types of units: A, T, G and C. These letters represent the type of base each nucleotide unit carries: adenine, thymine, guanine and cytosine. Here in this figure, you can see the base pairing in the Watson and Crick model of DNA. Here 2 hydrogen bonds connect thymine to adenine, 3 hydrogen bonds connect guanine to cytosine. The sugar phosphate backgrounds, which are depicted in grey color, run anti-parallel to each other, so that the 3' and 5' ends of the 2 strands are aligned. The structure of the bases as shown in this figure are the common, most stable forms in which adenine always pairs with thymine and guanine always pairs with cytosine. Here in this figure, you can see the base pairing of normal nitrogen bases in DNA. Shargaf's rule of DNA composition. The DNA molecule is made up of very long chains of the 4 bases: adenine, cytosine, guanine and thymine. In 1950, Erwin Shargaf published a paper stating that in DNA of any given species, the ratio of adenine to thymine is equal, as is the ratio of cytosine to guanine. This is known as Shargaf's ratios, and it was a crucial clue that helped solve the structure of DNA. Shargaf's ratios are universal. All forms of life obey this rule. Only the balance of AT pairs and CG pairs varies between species. DNA has equal number of adenine and thymine residues, that is A is equal to T, and equal numbers of guanine and cytosine residues, that is G is equal to C. This is the Shargaf's rule of molar equivalence between the purines and pyrimidines in the DNA structure. The correct structure of DNA was first deduced by J.D. Watson and F.H.C. Crick in 1953. When Watson and Crick described the double helix structure of DNA and proposed its semi-conservative replication based on specific base pairing to explain the faithful transmission of genetic information from generation to generation, they also proposed a mechanism to explain spontaneous mutations.